Okay, and so with that, I would like to introduce our next speaker, Avery Russell, whose talk is going to be on getting salty. Salt addition enhances the quality of low quality nectar.
Fantastic. Well, I want to thank Beacon for organizing this session and thank you all for your attention. I also want to first acknowledge that at Missouri State University, we live and work on unceded traditional territory of the First Nations, the Kickapoo, the Sioux, and the Osage. And if you're a student or have students or want to collaborate, please consider the Russell Lab. We work with bees, cognition, flowering plants, and their microbial mutualists. And I promise the puns aren't too insulting, although this talk might be. So without further ado, let's get to the science. Animals are frequently sodium limited, and this is a problem because sodium is needed for many basic physiological processes. For instance, nerve action potentials, proper muscle function, and transport of glucose, and all these kind of processes require sodium. As a result, many animals seek out sources of sodium from diverse sources. I'm looking at you, Salt Bay. For instance, animals often congregate on salt licks, or drink the sweat of others, or even the tears of various critters. And they even selectively browse for foods with higher salt content. I'm pretty sure also this must count as a salt. So if you have a sudden intense urge to lick the Himalayan salt lamp, maybe you're just sodium deprived. Sodium deficiency is especially common for pollinators like bees, which may often possess special physiological adaptations to recover sodium. These insects, of course, get the vast majority of their food from plant floral rewards. Plants, however, keep their sodium to themselves, and nectar in particular is quite low in sodium content. In the only 3 published surveys that we could find, the average salt concentration across 42 plant species from 29 plant genera across 15 plant families was only 0.04%. But there's also very high variation in sodium concentration in nectar among plant species, at least 3 orders of magnitude. And in fact, sodium concentration varies from being around the average, like this Aquilegia, to about 0.001% in this paintbrush, to about 2% in this hedge nettle. Given that pollinators are typically sodium limited, increasing the salt content of nectar could enhance the quality of that nectar. Accordingly, nectar salt content might manipulate pollinator foraging behavior, such as preference or constancy. For instance, pollinators might consider salty, low sugar concentration nectars just as good as saltless, sugar-rich nectars. So today, we focus on how the addition of sodium chloride to artificial nectar influences bumblebee nectar foraging behavior. So first, we assess how the presence or absence of salt in nectar affects preference for flowers with low versus high concentrations of nectar sugar. Second, we examine how the presence or absence of salt in nectar affects patterns of movement between flowers with low versus high concentrations of nectar sugar. And I want to emphasize this was work in my lab led by a visiting former PhD student, now Dr. Amanda da Silva, who is the first author and an expert in plant herbivore pollinator mutualisms and conflict. She successfully defended just a few months ago and is looking for postdocs. So if you have something available, let's get y'all in touch. So to achieve our objectives, we used bespoke artificial flowers and the common eastern bumblebee, which is just the bombus. This study consisted of 2 treatments, and these differed in terms of the nectar sugar concentration. In the first treatment, all flowers received 3 microliters of 50% sucrose. So that we could assess how preference and flower switching behavior differed across visits, we created 2 subtreatments. In one subtreatment, blue flowers received 0.1% salt in their nectar, and in the other, yellow flowers received the salt. In the second treatment, all flowers received 3 microliters of 20% sucrose, and again, we had 2 subtreatments differing in terms of which flower color offered salt. We started each behavioral trial with a fresh 5x4 array of artificial flowers, and we alternated trials by treatment and subtreatment to control for effects of time and day. In a given trial, a single bee was allowed to make up to 60 flower visits, after which the bee was euthanized so that it could not return nectar with or without salt back to its colony. For each visit to a flower, we recorded landings, which involved rejecting the flower without tasting or drinking. Oops. We also recorded landings and drinking from the flower, which involved the whole droplet being consumed, and landings and tasting nectar, which involved a less than 1 second probe of the droplet with the proboscis. So across all visits, grouped by bee, we examined preference for landing and drinking versus landing without drinking or tasting, landing and drinking versus landing and tasting, and finally, landing and drinking versus landing with or without tasting. We reasoned that when nectar quality was high, salt content would make little difference to bees. The excellent sucrose concentration should override most interest in the micronutrient. However, when nectar quality was low, we predicted that bees should attend to salt content of nectar and should therefore reject unsalted flowers more often, either after landing, but before extending their proboscis, or after tasting. In summary. Additionally, how pollinators move among flowers is important for the movement of pollen and thus plant reproduction, and can be independent of preference. We therefore also analyze the pattern of switching among flower types across bees. So here, for instance, we see a case where there are 4 total switches. The bees here show a strong preference for bulu flowers. In a different scenario, we also count 4 total switches, but in this case, preference for blue flowers is weak, and so we see that the proportion of switches does not necessarily correspond to the strength of preference. We therefore predicted that when nectar has high sucrose concentration, bees will switch indiscriminately between salty and unsalted flowers. Furthermore, we figured that when nectar had low sucrose concentration, bees should stay true to salty flowers for longer if salt improves the quality of the nectar reward. So, onto our sodium cool results. The following figures will all be laid out in the same way. We'll have the type of behavior we're examining on the y-axis, the treatment on the x-axis, either the low or the high sucrose treatment, and within each treatment we'll have means plus or minus standard errors coordinated with whether the flower received 0.1% salt or not. And the results of the generalized linear mixed effects model will be shown to the right of the graph. So, what did we find? Here we examine the proportion of landings that involve drinking nectar versus leaving without drinking, and we find that bees reject flowers with unsalted nectar much more frequently than flowers with salted nectar, but this preference only becomes very strong when the sucrose concentration of nectar is low. And in fact, bees were more than twice as likely to reject unsalted versus salted artificial flowers that had low sucrose concentration. Likewise, here we examine how frequently bees land and reject the nectar after tasting it versus drinking all of it. And we find that bees were around 3 times more likely to reject nectar without salt than with salt, but only when sucrose concentration was low. Altogether, we find that bees prefer to drink from flowers with salty nectar, and this effect became much greater when the nectar quality was low, such that bees were almost twice as likely to drink from flowers with salty nectar. Finally, we examine how frequently bees switching between flowers with and without salt. We find that nectar quality also affects flower switching, such that bees are much more likely to switch when flowers are saltless and low quality, but that this pattern is reversed when nectar quality is high. Now for some hopefully pretty sweet and savory conclusions. First, we show that bees overall preferred sodium in nectar, which agrees in broad strokes with what Van Valkenburg and colleagues reported in a field study using live flowers last year. And although 0.1% sodium is at the upper end of what flowers secrete, we hope our work will precipitate new studies, particularly given the limited data on salt concentrations in flower nectar. And second, we find that salt content especially improves the quality of low sugar nectars. And given that salt and sugar concentration in nectar varies considerably within and among species, we might expect pollinator-mediated selection on salt and sugar concentration simultaneously. And third, we find that salt content and sugar concentration oppose each other in affecting how bees switch among flowers, which could have consequences for pollen movement and thus plant reproduction. So our results suggest that flowering plant species should either offer high sugar concentration and no salt, or low sugar concentration and salt to promote pollinator movement among conspecific flowers. And with that, I'd like to thank you all, our funding, our support, and our advice. And here's a rapid fire look of how we refill these arrays during a trial. This is a different project as I lost my top-down videos of the salt experiment and the phone failure, but still wanted to show you. And with that, I'll turn it over to questions.